So proteins are a major topic in the biochemistry option, and that's because proteins are incredibly important and they do just about everything inside the human body or inside of other bodies. So it's a giant category of biomolecules, and there's a vast array of functions that they undergo. One of the most important of which is that they act as enzymes, which are biological catalysts, but they also give structure in the body. They transport things around. They help communicate in the form of hormones. They're part of your immune system, and you can break them down for energy, although that's really not their primary function in the body. Just some very simple general overview is that they are really big molecules. They're macromolecules, molecular masses in the thousands, and they are polymers, and really they are polypeptides, meaning they contain many peptide bonds. The monomer of a protein is an amino acid. It's called an amino acid because it has an amine group and because it has a carboxylic acid group. So the general formula of all amino acids is that it has NH2 attached to carbon with a hydrogen off of it, and then off of that carbon also is a carboxyl group. So every single amino acid has those three pieces in common. And then this square here, this R, or the R group, is what is different between each of the 20 common amino acids. So you could condense it down to here, and we're going to refer to this guy as the R group. So the specific type of covalent bond that links these different amino acids together when proteins are formed is an amide or a carboxyamide functional group, and it's usually just called a peptide bond in biology. And as we're going to see in every one of these subtopics of biochemistry, whenever we're linking two things together, that reaction is going to be the condensation reaction. So we're going to pull out HO from one molecule and H from another, and so that's going to form water as our byproduct, and we're going to form a peptide bond. So the peptide bond, if you want to look closer at it, this is the actual covalent bond that we're talking about, the bond between the nitrogen and the carbon here. So that's this nitrogen here and this carbon here, and this is the water that is removed. So the water is removed as a byproduct, and now you've got this is the peptide bond, and this whole functional group here is called an amide or a carboxyamide. Okay, so the overall general equation is that water gets removed, and now we've got this written here. This is amino acid number 1, amino acid number 2, so I've just said R1 and R2, and we're going to pull out one of these H's from the N, and we're going to pull off the OH from the carboxyl group there, and so this is the amide link on the right. So proteins do tons of different things, and so we're not going to cover everything about proteins in one video. The focus of this video is really going to be to look at amino acids. We're going to look a little bit at how they bond together in the different combinations, but then we're going to look specifically at amino acids themselves and some of their different properties. So the reason why amino acids are such good monomers in these gigantic polymer chains is that they have two functional groups that can undergo condensation. So if you think about this amino acid in the middle, it can condense with its amino group with an amino acid, another amino acid if it lines up with that carboxyl end, but its carboxyl group on the other side can then line up with the amino group on the next amino acid, and so it can form links on either side, and so that's why it's successful as a monomer in these condensation polymers, which we call proteins. So it has two functional groups that can condense, and that's why it can work as the monomer. Some very short-stranded things, and these are the types of things that you'd be asked to draw on the IB exam, are dipeptides and tripeptides. They could also be called dimers and trimers. So dipeptides are when two amino acids bond together, forming one peptide bond. Two amino acid residues, they might be called, but really think of it as two amino acid molecules condensing together to form one peptide bond, removing one water molecule. For any two amino acids, there's two possible dipeptides that can form, and that's because of that fact that the amino acids can bond on either side. They can bond on the amino side or the carboxyl side. So in this one, you'll see that the R2 amino acid still has its carboxyl group sticking out, but in the other version of the dipeptide, the R1 amino acid has its carboxyl group sticking out. So even though they're very similar, they're not exactly the same molecule that's been formed, this dipeptide, because of which functional group remains. So this is the N terminus or the amino terminus end, and you can see that the R groups are different depending on how they get stuck together. In a tripeptide, that's what it sounds like. It's going to have 3 amino acids in it, and so there's going to be 2 peptide bonds, and you got R1, and you got R2, and you got R3. So I'll put a little 1 there. That's really all there is to it. A question for you to try to figure out is how many different combinations can you form? How many different tripeptides can you form from just 3 amino acids? So when we had 2, there were 2 possibilities. We R1, then R2, or R2, then R1. Well, with 3, it gets a little more complicated. So if you want to pause the video, I'm not going to do this right now, but see if you can figure this out before class. How many different combinations are there for glycine, alanine, and arginine? Those are 3 examples. You can use their little symbols here to try to figure out the answers. Now, what's a protein? Well, a protein is called a polypeptide. So when you get more than 3 amino acids, now you're getting into that polypeptide territory. And usually proteins contain hundreds of amino acids, and so they're just absolutely gigantic, complicated molecules. These are some other practice problems for class, drawing dipeptides and tripeptides, so we're going to skip over that for now. The amino acids, one of the important things to consider about amino acids, one of the ways that they are often classified is based on their polarity or their water solubility. And really it comes down to the R groups because all amino acids have the same things in common, the amino group, the CH, and the COOH carboxyl group. So this is table 33 of the data booklet, and you'll see that, yeah, every amino acid has this part in common. So that's not really what we're going to be focusing on if we're talking about polarity or solubility. What we're going to focus on is the R group. So like for alanine here, the first one, its R group is a methyl group. Methyl is just C and H, and you should have a pretty good idea by now that that is completely nonpolar. And so alanine is not particularly water soluble, or at least compared to some of the other amino acids, it's not going to be that water soluble. If we look to the next one, arginine, it has some N to H's, so it does have some hydrogen bonding capable things, but it also has a pretty long carbon chain here. And so, that combination means it's maybe a bit more polar than the alanine, but not really, really polar. So if we go down the chain looking for ones that are more polar, well, this one's going to be a little bit more polar. It's got a couple groups that can hydrogen bond and a short carbon chain. So I think you get the idea. A lot of the ones on this page that I've shown are fairly nonpolar, pretty long carbon chains, but we've got a good hydrogen bonding carboxyl group here. We had another one up here. So there are some here that would be fairly polar, and then there are some that would be completely nonpolar, such as isoleucine, all hydrocarbon, and leucine here, which is all hydrocarbon. So you guys should be pretty comfortable identifying polarity at this point, and so just be prepared to do so when looking at these amino acid structures. Note this is only half of table 33. We're going to look at the other half in a second. Probably the most important structural thing to be able to talk about and figure out with amino acids is how their structures are going to change when the pH changes. It's going to be a little bit odd at first, but if you remember your acid-base chemistry, this should make a little bit of sense. An amine group is really a base. It's a proton acceptor because the nitrogen atom has a lone pair of electrons, which is fairly good at taking protons away from other things. So think of ammonia, think of amines. So the amine group is a weak base, and the carboxyl group is a carboxylic acid. So you should think of this group as being a weak acid. So you've essentially got something that is both a weak base and a weak acid. So Well, first of all, if you're in the HF class, that should remind you of a buffer. And indeed, amino acids can act as buffers, and we'll talk about that in more depth. But also, if you put an acid and a base in solution together, well, they'll react with each other. And that's actually exactly what happens. If you put amino acids by themselves into solutions, this proton from the carboxyl group will actually get donated to the amine group. So it actually undergoes an acid-base reaction with itself. And you form this structure on the right. So notice the proton has moved to the nitrogen over here. This is the nitrogen atom. And so now that H plus has given the amine group a plus charge because now it's NH3 plus. And over here, this oxygen lost its hydrogen. And so now this O right here has a minus charge. So in solution, amino acids can form a double ion, something we haven't really seen before, that is called a Zwitter ion. Now, notice that because it has a positive and a negative charge, overall, it actually has no charge. So it's very, very odd. It's a double ion that has overall no charge. That is the Zwitter ion. Now, when the heck will this amino acid actually exist like this? Well, it turns out that for every amino acid, there is a particular pH value when it will exist in this form. And that is known as the pH of its isoelectric point. Think about that term, iso. We've used that term a lot with isomers and isotopes. It really means same. So the isoelectric point means when you're at this point where there's no effect from electricity. So when you're not like thinking, if you think of isotonic and hypertonic and hypotonic. Isotonic is when there's no change, there's no effect because concentrations are the same. Being isoelectric means that there's no effect from electricity. So those values are listed in the data booklet. So here's the second half of table 33. And so along with the structure and the name and this three-letter symbol, they also list the pH of the isoelectric point. So these pH values are when these different amino acids will exist as that double ion, the Zwitter ion. Okay? And so the pH values can range a fair bit, but really it depends on what the R group is. So if R groups contain a second carboxyl group, so every amino acid has a carboxyl group, but sometimes the R groups contain one as well, that's going to make those amino acids more acidic. And so their isoelectric points are quite low. They're much lower than 7. If your R group contains amine functional groups, amines are weak bases, and so that tends to make the isoelectric point quite high. And if your R group just contains like hydrocarbon or something that's not acidic or basic, then the value tends to be around 6, somewhere in the 5 to 6 range. Okay, so we can look here, and you'll see when there's an amine, right, the isoelectric pH tends to be quite high, associated with base. When you have one that is, let's see, I don't think we have any here that have acid groups on them, and you'll note that these are all really around the mid 5s to 6, because these all have pretty much non-acidic or basic functional groups. But if you look back at the other part of the table here, these ones that are called like aspartic acid or glutamic acid, they have quite low isoelectric pHs, which you'll see if you look in the data booklet. So one of the things that you're going to be expected to be able to predict is what's going to happen to the Zwitter ion, or what structure will the amino have in general, depending on the pH value. So you'll have the data booklet on paper 3, and so it's pretty easy to know when it's going to look like this. This is the Zwitter ion form. It will look like that at that pH value that's listed in the data booklet. But what if the pH is lower than that? So if the pH is lower, that's kind of like saying that you're adding acid. So you should associate acid with H+. And if you think about this structure here, there's a positive charge on this end, there's a negative charge on this end. If you put this amino acid into a solution with lots of H pluses, try to think, where are those H pluses going to be attracted to? What is going to happen? I will give you a second to think about it. Well, one of the fundamental rules of this class is that opposites attract. So these protons, if you put an amino acid into a solution with extra protons with a lower pH, well, the protons are going to go towards the negative charge. And these two things are going to bond together. And so when you put a Zwitter ion or an amino acid into a low pH solution, it's going to end up with an overall positive charge because this H plus is going to bond on to this oxygen. We're going to reform our COOH carboxyl group, and then this positive charge will remain, and so overall the amino acid will be positive. If you put a Zwitter ion into a basic solution, a solution with a higher pH than its isoelectric point, you should probably associate base at this point with hydroxide ions. So what's going to happen? Which part of the amino acid will this be attracted to? Well, it's going to be attracted to the positive part. So this OH and this H plus are going to react to form water. So you're going to pull off that extra proton that was on the amino group. And now you'll have NH2 on this side because one of the protons was lost. But this side will remain as COO minus. And so when you put things into basic solutions, and the end result is that they end up with an overall negative charge. So it should be fairly straightforward. You put things into acid, think H plus, the amino acid will end up with a plus charge. If you put things into a basic solution, OH minus, the amino acid will end up with a minus charge. And if you put things into the pH right at their isoelectric pH, they will exist as the double Zwitter ion. Last topic for the SL, I think this is still in the SL syllabus, so I'm going to just talk about it briefly. I mentioned before, amino acids can act as buffers. That means they can neutralize both acids and bases if small amounts are added to them. And it's because they have those two functional groups. So if you add base, if you add OH minus, there are lots of groups, depending on the form of the amino acid, that can react with this OH minus. So this H plus here we talked about could react with it. This H plus here when it's in Zwitter ion form, and this carboxyl H here, if it's in the positive form, all of these H's are acidic and can react with the OH minus. If you add acid to an amino acid, well, it can react with the carboxyl minus ion in that form. And if you're looking in a high pH, then it can react with either the nitrogen here or this carboxyl minus here. So because amino acids contain both acidic and basic functional groups, they can neutralize both acids and bases to a small extent, so they act as buffers. So in this video, we talked about proteins being polymers of amino acids, how they are amphoteric, and we just talked about that, and how they can exist as Zwitter ions, cations, and anions. We've also talked about how you get structural formulas when they condense together. Hydrolysis reactions would just be the reverse of condensation reactions, so cutting dipeptides and tripeptides apart to reform the amino acids. We talked about their solubilities. Melting points would be similar to solubilities, thinking about the IMFs of the R groups and how that would rank them for attractions and therefore melting points. And finally, talking about how pH change will affect their charge. So that's what we talked about in this video, and obviously we'll review it in class.